I'm Smitha Iyer. I'm an assistant professor at UC Davis. My lab studies T-cell responses to viral infections, and I work at the Center for Immunology and Infectious Diseases and the California National Primate Research Center. My lab is at the Center for Immunology and Infectious Diseases, or CIID, which is adjacent to the CNPRC, and it was thanks to the strong collaboration between these centers that we were able to catalyze COVID-19 research at UC Davis. For instance, both the CIID and the CNPRC have access to biosafety level 3 facilities, which are critical and the only way to do COVID research. We have highly trained staff as well as scientists that are able to ask the right questions and the resources to find the solutions. And of course, last but not least, we have highly proactive leadership that was able to quickly get these studies going and really set the stage for COVID research at UC Davis.
I'm John Morrison. I'm director of the California National Primate Research Center and professor of neurology here at UC Davis. As with all 7 primate centers, we have been very involved with COVID research over the last year. We are testing antibodies, monoclonal antibodies. We are testing vaccines. We're testing other therapeutics. We're also developing the monkey model so that we can understand the pathology to a greater degree than you could possibly do in humans. And we've now moved into the brain in our COVID research, and we'll be able to determine why so many patients end up with neurologic complications.
The CNPRC is unique among the 7 national primate centers in that we have access to what is perhaps one of the most well-characterized geriatric colony. We know that one of the critical factors that increases the severity of COVID-19 infection is in fact age and type 2 diabetes. We know that these factors also underlie the efficacy of vaccines. So our ability to control for and then consequently study these factors is very important if we are to fully understand the pathogenesis and design better vaccines and make better therapeutics. The fact that we have a vaccine pretty much within a year following infection is because of the large investment in biomedical research that has predated SARS-CoV-2. This included research in viruses not related to the coronavirus, such as HIV, the Zika virus, RSV, and of course SARS-CoV-1. The work that we do here really lays the foundation to understanding the virus and also the immune response and the host response to the virus. And as you know, in order to defeat the enemy, you should know the enemy. And the more we study this virus in an appropriate model system, the better we are to design therapeutics as well as vaccines against future viruses.
We also have a very well-developed capacity to move from infectious disease to other areas of the primate center. For example, we were able to move very smoothly from the infectious disease elements of Zika and COVID into the lung and into the brain. And we now are doing very detailed neurobiological studies of animals that have been exposed to COVID. If we had steady support from donors, the most important thing we'd be able to do is to develop novel models of human disease. NIH is reluctant to support the development of a model. They'd rather support studies on that model. But the most important thing for us to do is to develop new, powerful models of human disease.
Donor support would have a significant impact on infectious disease research at the CNPRC by giving us access to cutting-edge technology and equipment such as flow cytometers to study immune cells, microscopes to study tissues, and instruments that allow us to study cells at a single cell level. By using this cutting-edge technology, we can gain better biological insights into infectious diseases and advance the scientific mission of the CNPRC, which is to improve human health.